In recent years, researchers have begun to grow tiny organs from human stem cells to get better view of development and disease. The latest organ to get this treatment is the brain, after miniature versions of our most complex body part were produced in a lab. And these mini brains have already proved their worth by confirming how the Zika virus ravages fetuses. The lab-grown mini brains, which researchers say are truer to life and more cost-effective than similar research models, have been developed at Johns Hopkins University in Baltimore. Though the organs themselves are about the size of a pinhead, the bioreactors where they grew were comparatively large, about the size of a drinks can. This made working with previous versions of mini brains expensive, given the high cost of the nutrients needed to cultivate human stem cells in the lab. To solve this problem, a trio of high school students devised a way to make the equipment for growing the mini brains with a 3D printer. The students had worked with 3D printers and thought they could be the key to producing a better bioreactor, one that would fit over commonly used 12-well laboratory plates and spin the liquid and cells inside at just the right speed to allow the cells to form the miniature organs. The group has so far used the new bioreactor, dubbed Spin, to make 3 types of mini brains mimicking the front, middle and back of a human brain. They used the forebrain, the first mini brain with the 6 layers of brain cell types found in the human cortex, for the current study on Zika. We have been working for 3 years to develop a better research model of brain development, and it's fortunate we can now use this one to shed light on the major public health crisis posed by Zika infections, said Dr. Hongjin Song, professor of neurology and neuroscience at the Johns Hopkins University School of Medicine's Institute for Cell Engineering. This more realistic, 3D model confirms what we suspected based on what we saw in a 2-dimensional cell culture, that Zika causes microcephaly mainly by attacking the neuroprogenitor cells that build the brain and turning them into virus factories. One thing the mini brains allowed us to do was to model the effects of Zika virus exposure during different stages of pregnancy, added research partner Guo Liming. If infection occurred very early in development, the virus mostly infected the mini brain's neuroprogenitor cells, and the effects were very severe. After a while, the mini brains would stop growing and disintegrate. At a later stage, mimicking the second trimester, Zika still preferentially infected neuroprogenitor cells, but it also affected some neurons. They found that growth was slower, and the cortex was thinner than in non-infected brains. These different effects correspond to what doctors have seen in babies of women who contracted Zika during pregnancy, as well as miscarriages. And the earlier in pregnancy Zika infection occurs, the more severe its effects. According to the JHU team, the next steps will be to test drugs already cleared by the Medicines Authority on the mini brains. Through this method, they hope that one of the treatments might provide some protection against the effects of the virus. In addition, they have included plans for the spin with their paper, so other researchers can produce the bioreactors themselves. Beyond the immediacy of the Zika threat, Song explained that the approach could even be used to grow replacement brain cells to replace those damaged in conditions such as Parkinson's disease. Professor Song concluded, "This is the next frontier of stem cell biology.